Hello everyone, welcome to One Academy. So in this lecture I am going to talk about the cell cycle. What is cell cycle? It is a sequence of events that would lead to the cell division. Now when we talk about cell division, we basically talk about the various components inside the cell dividing. The components of the cell are basically the genetic material and the other constituents. Other constituents are the cell organelles. So if we define it, it is defined as sequential events, that means a step-by-step process by which a cell duplicates its genetic material, synthesizes constituents of the cell and eventually divides into two daughter cells. So from a single parent cell we get two daughter cells. Now this process of cell cycle is variable from organism to organism. For example, a yeast cell divides once every 90 minutes, but a human cell takes approximately 24 hours. So there is a large difference between the rate of division in case of different organisms. Apart from this, it is variable from cell to cell. For example, inside the human only, there may be some cells that have a fast rate of division, while there might be some others which would have a relatively slow rate of division. So this process of cell cycle is not constant for the whole set of organisms, rather it is variable for each. Now I am going to talk about the phases of cell cycle. There are basically two phases, the interphase and the M phase or the mitotic phase. Now we call interphase as a preparatory phase. There is a reason for doing so. Preparatory phase is because here preparation for the process of cell division is occurring. Here no actual cell division is happening, but the preparation is done by the cell in order to start the cell division. What preparation? The replication of the genetic material, the duplication of its cell organelles, and the synthesis of various biomolecules like proteins, RNA, etc. It is also called as the biosynthetic phase because here synthesis of cell organelles, genetic material and biomolecules is occurring. Now the M phase or the mitotic phase, which is called as a multiplication phase. Now why do we call it a multiplication phase? Because one parent cell would give rise to two daughter cells. So in a way the parent cell is multiplying. That is why we refer to as multiplication phase. Now I will give a brief discussion about the interphase and the M phase. So what is interphase? I already told it is a biosynthetic or the preparatory phase. Now we also refer it to be resting phase. Resting phase because here no actual cell division is occurring. So resting with respect to the cell division process. But that does not mean that the cell is completely inactive during this phase. Cell is stocking all the biomolecules which would be required during the cell division process, and the cell organelles are being synthesized. So it is not an inactive phase, rather it is a metabolically active phase. Okay, so we call it resting with respect to cell division because no cell division is occurring. We call it as resting phase. Now interphase, as you already know, involves the replication of the genetic material and synthesis of cell organelles and biomolecules. Now this phase has been divided further into G1 phase or the gap 1 phase, S phase or the synthesis phase, and gap 2 phase or the G2 phase. Why do we call it as G1 phase or gap 1 phase? Because it forms the first gap between the M phase, which is the multiplication phase, and the S phase, which is the synthesis phase. Now S phase is called as synthesis phase. Why? Because synthesis of genetic material is occurring during this phase. And then we have the G2 phase or the gap 2 phase, which is a second gap between the S and M phase. So G2 is similar to G1, but the only difference is that it forms a second gap. The gap is between S and M only in both cases, but it is the second gap. Okay. Next we have the M phase, which is the actual cell division phase. Here the cell division is occurring. Now the two processes that are involved in the M phase are the karyokinases and the cytokinases. Karyokinases is composed of karyon, that means nucleus, and kinases, that means breakdown or division. So it is nuclear division process. Now this process is a complex one which further involves subphases like prophase, metaphase, anaphase and telophase. These phases will be discussed in detail in subsequent presentations. The next is the cytokinases. Cyto comes from cytoplasm and kinases again is breakdown. So here the division of cytoplasm occurs. Finally, two daughter cells are formed. And therefore, because all the components of the cell are being divided or separated, we call it as a reorganization and separation phase. The M phase is referred to as reorganization and separation phase because the cell components are being separated and reorganized among the two daughter cells. Now this is a diagrammatic view of the cell cycle as a whole. The cell cycle starts with the G1 phase, which is the gap 1, first gap between the M and S phase. Now some of the cells, they don't divide. They would enter a phase called as a G0 phase. Here these cells would no longer divide, but they would be growing. Once they have grown, they would no longer divide. They'll only remain metabolically active. They would not divide further. So they enter a phase called as G0 or quiescent phase. In humans, nerve cells are such cells. They won't divide once formed until and unless there is any cell death or cell injury. Okay, so after G1 we have the S phase, that is the synthesis phase where the genetic material is synthesized. Next we have is the G2 phase, which is the gap 2 phase, and here is the second gap between the M and S phase. Finally, we come to the M phase, which is divided as cytokinases and all karyokinases. Okay, so I hope you understood the basics of cell cycle. Thanks for watching.